Hi there. In this video I'll talk about DNA mutation first and then talk about repair, and then in a subsequent video I'll talk a little more about repair, specifically with regard to recombination. Biological evolution has been primarily influenced by three main factors. One is mutation, mainly of DNA organisms, but also RNA viruses can be included in this. And RNA viruses, of course, mutate at a much faster rate. But I'll talk mainly about DNA in this video lecture. Second is selection at various levels, sort of selecting out which mutations are either advantageous or disadvantageous. And thirdly here, that also affects evolution, is random genetic drift. That is, mutations that either have no positive nor negative effects on the organism, such that those can build up, and if the organism moves to a different environment or whether the environment changes, then those mutations can be subjects of selection. So evolution is dependent on these three factors, and you want some mutation, of course, to create a diversity of genotypes for selection in different environments. So that's absolutely important, but you don't want so much mutation that they cause mainly negative effects on the organisms. So what has evolved is a balance between having enough mutations to maintain a diverse population of genes, essentially, versus too much mutation, which causes more extinctions, so that those organisms no longer survive. So there are not only mechanisms for mutation, but also correction mechanisms that I'll talk about later in this video, and those are repair mechanisms. So before I get into mutation, I want to just sort of introduce a way of thinking about mutation in a more complete way. So most people talk about selection as acting on the organism as it pertains to its external environmental challenges, and so that's mainly what the mutation rates for a particular gene or a particular character are normally dealing with. However, there are selective pressures that exist at all levels within organisms. So the selection exists at the level of transcription. Is the gene transcribed or not due to that mutation? Splicing, is there still accurate splicing? Transport out of the nucleus, translation, etc. That is intracellular selection, but there's also intercellular selection among the cells in that population, as well as selection at the tissue level and all the way out to the whole organism. And then you get to these extra-organismal level selections, which are the things that are normally discussed when you talk about selection. The total of all of those, so you look at the mutation rate for specific genes and specific nucleotides. So selection at all those levels is taking place so that you have an overall mutation rate that doesn't necessarily reflect what the mutation rate actually is in that particular gene or nucleotide. So I'll try to illustrate that here. So if you have DNA within a cell, then you can have DNA changes. That is, a change from an A to a G or a T to a C or something like that. They're not considered mutations unless they're inherited. So the definition of a mutation is if it's inherited. So there's this inheritance filter between just a simple change and a mutation that's carried on throughout the population of those cells. If there's a change up here and it doesn't become a mutation, then you have it filtered out at this point. So either that change is then repaired by the repair system, or that cell doesn't divide. So those are two ways that you can have filtering or selection at this level. At the next level, the question is it transcribed or not? If it is transcribed, then you get an altered RNA. If it's not, then this transcription is stopped at this point, and you have selection at the transcriptional level. So if it is transcribed into this altered RNA containing a mutation, does that mutation affect splicing or transport out of that RNA out of the nucleus? So then there's a processing filter that controls what RNAs are never allowed to leave the nucleus. So the next question is, does that altered RNA that has now left the nucleus, is it possible for that to attach to a ribosome and be translated? So then there's a translation filter. If it is translated into a protein, many proteins then have to be either transported to a particular place in the cell. They also have to sometimes be changed or phosphorylated or chopped in some ways. Further processing is necessary for them to function. So there's then a processing filter. Is that altered protein able to be processed properly? And then if it is processed into a mature protein and it's transported to the right location in the cell, is it actually a functional protein? So it could be processed in the right way, transported to the right place, but the mutation affects somehow the folding or the activity of that protein. So there's another filter there, another selection filter. And so now we have a functional protein, but now that affects that cell or the whole organism in some way. And now there's finally this environmental filter. How does that protein affect the organism and its actions in that environment, in those cells, in that tissue, etc.? So all of these up here are then intracellular levels of selection, whereas this one down here, these are intercellular. That is, how does it interact with other cells around it? Extracellular, how does it interact with things more distant? And extraorganismal, how do all those cells together function for that organism in order for it to survive in that environment? And in most cases, people are measuring out at this level. How does that cell or that organism interact with its environment? Is it successful? Is it not successful? Partly successful? Whereas all of these mutations and these filters back here affect the success of this organism out here. And so often the ultimate mutation rate that is measured for a particular trait or gene is an underestimate of what's actually going on back here as far as the number of mutations that occur in that gene or with that particular nucleotide. So now let's look at where in the function of a cell these mutations can occur. So at the DNA level, then you can have things like mutagens in chemicals causing mutations. Also, recombination events can cause mutations in the DNA. Many types of microorganisms, bacteria, fungi, viruses, etc., can also cause changes to DNA. So those can be considered mutagenic. Simply replicating the DNA can also introduce mutations, and often they are introduced at that level. But microorganisms, again, can affect that process. Mutant replication proteins can affect that. Low nucleoside triphosphate concentrations can cause that. And then DNA polymerase slippage and dislocation can also cause mutations at that level. However, there are correction mechanisms, at least in DNA organisms. In RNA organisms, there aren't in the viruses. But that can also be affected by low nucleoside triphosphate levels, mutant repair proteins, again, microorganisms, and then DNA polymerase slippage and dislocation, because DNA polymerase is involved in the repair of DNA in almost every case. So here's a list of a few of the mutagens, and they include ultraviolet irradiation, some chemicals, ionizing radiation, infections, and some enzymes also can cause mutations in the DNA. As I said, replication itself can introduce mutations, and this is a common way for mutations to be introduced. One is by DNA polymerase itself making mistakes, and it does. Altered DNTP pools, primase and the RNA primers, gyrase and helicase, mutant enzymes such as those, and then other things such as single-stranded binding proteins and REC-A or REC-1, which are single-stranded binding proteins as well. All of these are required in DNA replication, and if there are either low levels of some of those or they're mutants themselves, then you can introduce more errors during replication. So there are also base pairs don't always line up exactly as we think of in the Watson and Crick sense, that is AT pairs and GC pairs. You can also have GT pairs, which happen quite often, AC pairs, which also occur often, and GA pairs, which occur less often. These can all be repaired by repair enzymes if the wrong base pair happens to be sensed by the repair systems. There are also problems with the repair enzymes and pathways occasionally. So you can have problems with the base excision repair system, the nucleotide excision repair system, mismatch repair system, DNA glycosylases, and also some others. So these are, if you have a mutation and it isn't repaired, then it can be a pretty big problem, actually. So this gets into what I was talking about, that you have to have mutation systems actually occurring. There have to be mutations in order for there to be enough diversity in the genotype or the genes or genomes of the population. But this has to be controlled somewhat by repair systems so that you don't have too many errors, which would cause too much damage to the DNA and lead to death and extinction. So there are certain rates of errors created by some of these different mechanisms. And so barring any mutagen or anything else, these are just natural things that are occurring in those cells. So just by mistakes in base pairing, the cumulative error frequency is about 10 to the minus 1 to 10 to the minus 2, which is 1 in 10 to 1 in 100. So it happens very frequently. But then you have DNA polymerase, which has a proofreading function, and you get that level down to 1 in 100,000 to 1 in a million base pairs is mutated. Accessory proteins that help this process of DNA polymerization, the replication, brings that down to about 1 in 10 million. And then post-replicative correction mechanisms bring that down to about 1 in 10 billion for each replication cycle. So in an organism with a 10 to the 10th base pair genome, humans are about 3.5 times 10 to the 9th. So I'm just putting this sort of on a human scale, but 10 to the 10th is easier to deal with than 3 times 10 to the 9th. So the cumulative errors then for that size of a genome, just on base pairing mutations, errors, then you'd have 10 to the 8th to 10 to the 9th errors per replication cycle. For DNA polymerase, so that has higher fidelity, you would still have from 10,000 to 100,000 mutations per cell division. And then accessory proteins would bring that down to about 1,000 mutations per replication cycle and division. But then if you add the repair mechanisms down here, then you get that down below 10 mutations per replication cycle. Still not zero. And remember, you don't want it to be exactly zero, because mutations got us to where we are now, right? The evolutionary systems got us to where we are now. So you have to have some mutation in order for evolution to occur. So how does this affect you or relate to you? In your body, you have about 10 to the 13th cells, about 10 trillion. Each of which was the product of a replication cycle ever since you were a zygote. So if there is, on average, one mutation per genome per replication cycle, then you will have approximately 10 to the 13th mutations scattered throughout your body in different cells usually. The probability of a mutation in a particular gene in a particular cell is about 5 to 10 in 1 million, a gene being about 1,000 base pairs, and the haploid genome of humans is about 3.2 billion base pairs. But the probability of a mutation in a particular gene in any of the 1 trillion cells is actually higher. So you'll have mutations, again, not being zero, are scattered throughout your cells and scattered throughout your genes. If the mutation occurs early in development, that is at the zygote stage or shortly thereafter, then that mutation will be inherited by more of the subsequent cells, and you'll have more mutations after that. So this is why people try to be very careful when there's an embryo or fetus, in that mutations very early in development will cause larger effects in that organism than mutations that are happening when you're 60 or 70 years old. So a mutation that occurs in the zygote, then you'll have that mutation in virtually all of the cells in your body. If it occurs after the first division, then it'll be in half of the cells in your body, etc. And so by the time you're a fully mature adult, then those mutations should have less effect on the overall development, at least. Now, you can still get mutations that cause cancer and other types of tumors later in life as well. So here I want to, this is just 2 base pairs, an AT pair and a GC pair, but I want to illustrate where some of the most common mutations occur in DNA. So one of the most common mutations, spontaneous mutations in DNA, are hydrolytic attacks. So this would be under acid conditions are one of the ways that you get hydrolytic attack. And it's primarily cutting off the purines. So you have then apurinic sites, apurinic nucleotides. So here's a DNA chain here, and then hydrolytic attack on here on this glycosidic bond, which is the bond between the base here and the sugar, that's called the glycosidic bond, would remove that adenine. So you'd have a T over here and no base over here, even though the structure of the DNA is still there. The same with guanosine. And to a lesser extent, you can get depyrimidization, forming pyrimidinic sites of a C or a T. There's also hydrolytic attack often here on the cytosine, removing that amino group. And then there are some minor effects too, where you can nick the DNA, that is, break the diphosphate bonds, so that you can essentially nick the strand of the DNA. The next major type of reaction to DNA, to degrade DNA, are oxidation reactions. Now remember, organisms first evolved in a reducing atmosphere, so there wasn't a lot of oxygen around, so that, and other oxidizers, so that DNA first evolved in a reducing type of cell, a cell that was used to living under reducing conditions. Now we live under more oxidative conditions. So there are these reactions that occur with DNA. One is to break that sugar apart. Another is to act on this part of the ring structure in purines, specifically more so in guanosines than adenosines, as well as more so than in cytosines and thymines. But these will many times break ring structures apart, these oxidative reactions. Third set is indicated in green here, and those are S-adenosyl methionine transfers of methyl groups. This is actually an ancient way to transfer methyl groups and probably existed, in organisms 3.5 billion years ago. But it happens to be one that is damaging to DNA, although it's still used in other processes around the cell. This affects primarily the purine rings of adenosine and guanosine, which will attach methyl groups to guanosine and adenosine at those nitrogen positions, as well as a couple of other positions on the rings as well, but to a lesser extent. And you can get methyls also attached to cytosines and thymidines in this case, but as you can see by the arrows, that happens much less often. Mutations of bases don't occur all at the same rate, so all of these arrows here indicate the relative frequency that those mutations occur. So transitions, that is a change from a purine to the other purine, and changes of the pyrimidine to the other pyrimidine occur more often than transversions, which are changes from a purine to a pyrimidine or a pyrimidine to a purine. Then there's this C to T change, which you can see the arrow is much longer, indicating that C to T changes occur more frequently than T to C changes or any other mutation. And this is because often cytosines are altered to become 5-methylcytosine. So there's a methyl group on the number 5 carbon in the pyrimidine ring. This is done for a few reasons. One is to distinguish between the template DNA, which is methylated, and the newly synthesized strand of DNA that's being synthesized during replication, which isn't immediately methylated. So this way, if there's a mutation, then the repair mechanisms can tell which is the original template strand, which is the newly synthesized strand, and it will then alter the newly synthesized strand over any changes it makes to the template strand. A second mechanism is when there are a lot of methylcytosines in a particular sequence. It will twist the DNA somewhat so that you can actually form, instead of normal B DNA, it can be reversed to form Z DNA. And that normally in a promoter region will turn off transcription of that gene. And then a third way, function of the methylcytosine is to protect the DNA from restriction enzymes. So when they're methylated, then normally those nucleases don't recognize that DNA and will not digest it. And this occurs in bacteria as well as in archaea and eukaryotes. The thing is here that once you have a cytosine, you methylate it, and then if it becomes deaminated at this number 4 carbon position, then that becomes thymine, and that's indistinguishable from any other base that should be in a DNA, so that repair enzymes often miss that as being a mutation, and then that mutation gets copied into the new strand. So that's indicated here. If you have a cytosine and it's deaminated, it becomes a uracil. But repair enzymes recognize that uracil as being in the DNA, and it's not supposed to be in there. So it'll chop that uracil out, and the repair mechanisms, especially the DNA polymerase, will then fill that in with the C that was originally there. However, if you have a methylcytosine and that becomes deaminated, that's a thymine, and during repair and or replication, this strand produces a new double-stranded DNA that's like the original, but this one now replaces that G with an A, and so that becomes a mutation after division of that cell. Another very common mutation, in fact, whenever you go out into the sun or you sit under UV tanning lights, then the DNA is being changed, and unless it's repaired, then you will have a mutation. So this is the formation of pyrimidine dimers. So adjacent pyrimidines, in this case a T and a T, when exposed to UV irradiation, you'll have a butyl ring formed between these two carbons and these two carbons. And so it forms a pucker in that DNA chain that is usually recognized by repair enzymes. And the frequency is this. So thymidine dimers occur most often. Then T to C and C to T mutations or dimers occur less often, and C to C dimers occur even at a lower frequency than that. And there are people who have a disease called xeroderma pigmentosum, which is genetically inherited, and it causes a problem with the repair of these thymidine dimers, so that every time they go out into the sun, or even if they go into a room with fluorescent lights, there's enough UV radiation coming off those that they will develop lesions on their skin. So here are some of the more common base changes that occur in DNA and whether they are repaired in vivo or not. So deamination of a C, it becomes a U and it'll pair with an A, but there are repair enzymes for that. And as I just mentioned, a 5-methyl-C, when it's deaminated, becomes a T that still pairs with A, and there's no known repair of those. So those get through, and you have a larger number of C to T changes in those cells, or in any cell actually. Deamination of these purines, either A or G, become hypoxanthine or xanthine, but there are repair enzymes for those. Depurination by acid conditions, usually no base, and that becomes ambiguous, but those are also repaired. Oxidation of a guanosine produces an 8-hydroxyguanosine, which pairs with an A. And it's uncertain whether there are repair systems for those. Those happen most often in mitochondria that are exposed to higher levels of oxygen in the cell because of oxidative phosphorylation. Adenines often become cyclic. That is, it's a cyclic oxidation between part of the base and part of the sugar to become 8,5' cyclic adenosine. And it's sort of ambiguous pairing also, and there are no known repair systems for that, so those become fixed. And then there are pyrimidine dimers, primarily TT dimerization, and they don't really pair with anything. They cause a pucker in the DNA chain, but those do have effective repair mechanisms for those. And then hydroxymethylation of any of the nucleotides, and it's unknown what those will pair with and if there's any repair of those particular mutations. So here I just want to go through some of the common types of mutations and their names so that you know the names of these mutations. So here's the original sequence at the top, and then if you have a transition, there's a C to T change in this case for that transition. So a substitution of one pyrimidine for the other pyrimidine. And here's an example of a transversion of a pyrimidine being substituted for a purine, so a G to C change in this case. Here's a deletion of that ACCTA sequence. Often these are a product of recombination, either by looping out of part of the chromosome, and then you get a recombination event deleting that little circular region, or two chromosomes misaligned, so one actually has a deletion and the other one receives an insertion. So those are a couple of ways that you can get those. And so here's an insertion simply by inserting this AAAGC sequence in between the G and T of the original sequence. Here's an inversion, and it might look a little strange at first because the sequence has changed, but remember, during an inversion, you're dealing with a double-stranded molecule. So this is essentially inserting the opposite strand in the reverse sense into this sequence. In many genes, that is ribosomal RNAs, tRNAs, and small RNAs, those mutations are just transcribed into the RNAs that then, so they may affect that RNA in certain ways, but in mRNAs then, of course, there's a code, a genetic code, so you can change one of the nucleotides in the triplet, and that can either change what tRNAs are used or change the amino acid that's encoded there. So in the case of a synonymous mutation, here's a GTC changing to a GTA, and both of those encode valine. So a valine amino acid would be inserted into the amino acid sequence at that point. While that doesn't necessarily change the sequence of amino acids in the polypeptide, depending on how available those particular tRNA anticodons are, it may affect the speed or something else about the production of that particular protein. So they're not necessarily innocuous mutations. But then there are missense, also called non-synonymous mutations, especially changes in the first and second nucleotides of the codon. Here, there's a GTC to a TTC change, and that will change a valine to a phenylalanine. That likely would have an effect on either the folding or the function of that protein. These nonsense codons usually greatly affect the polypeptide. So in this case, the AAG is changing to a TAG, and instead of adding a lysine, this terminates the chain, so the protein is terminated at that site because that's a stop codon. The nonsense mutations are associated with several types of diseases, one of them being muscular dystrophy. So there's a stop codon in the coding sequence that truncates the protein, and so the dystrophin protein is not formed, and that causes the disease muscular dystrophy. There are other diseases also that are caused by these nonsense mutations that cause a stop codon in the coding sequence. This is a summary of some of, but not all of, the repair system. So this is base excision repair here, mismatch repair here, nucleotide excision repair, and then I'll talk in more detail about recombination repair. So here's base excision repair, where there's a missing base or an inappropriate base placed in the DNA, And so that's recognized by glycosylases, which chop off those bases. And then a long section of the DNA is removed by specific enzymes. And then DNA polymerase comes through and polymerizes a new strand in that section. And then DNA ligase completes the process by ligating the last two bases together. The next repair system here is mismatch repair. So when you have mismatched bases here, an AC pair, then again that's recognized. Enzymes chop out the piece in actually more, sometimes 100 base pairs or more. And then DNA polymerase comes through, and then ligase again finishes that section by ligating the last two bases together. The nucleotide excision repair is the thing that recognizes the pyrimidine dimers and corrects those. So it recognizes that region, chops it out. DNA polymerase comes through and makes new strands, again, up to like 100 or so base pairs. And then DNA ligase ligates the last two bases together. I won't talk about recombination repair in detail here, but you can imagine how this occurs, where you get a damaged piece of DNA, and then you bring a homologous DNA next to that, and you can use recombination to essentially create two recombination events that then removes that part of the DNA that is mutated and inserts a new piece in there that is without the mutation. This occurs a lot in bacteria that are resistant to high amounts of radiation, such as Deinococcus radiodurans, which once the DNA is damaged, it comes in there with a lot of repair enzymes, more than we actually have, including a lot of recombinational repair, where out of a bunch of broken strands of DNA, it can piece those together into new pieces of DNA, new chromosomes that are fully repaired. Here's another illustration of some of those repair mechanisms, as well as another one. So the one that I didn't mention before is direct reversal. So there are systems in bacteria where if a mutation is discovered, then there's a removal of the offending base, the mutated base, and insertion of the correct base in that part. Mismatch repair also does that, but it chops out the entire piece, and then DNA polymerase comes in and replaces that. Nucleotide excision repair usually is caused by noting that systems noticing these bulges, such as in thymidine dimers and guanine dimers as well. Again, those will chop out an entire piece, and then DNA polymerase will come in and replace that. Recombinational repair here is by, of course, recombination or crossovers using a repaired strand or a fully non-mutated strand, and then crossing over to avoid inclusion of the mutated part of the DNA. Also, there's base excision repair here, where a base is noted, such as a uracil that's chopped off, and then an entire piece here is removed using endonucleases, and then DNA polymerase comes in and repairs that strand, and DNA ligase then puts together the last two nucleotides. So here's an illustration of base excision repair. If it recognizes a damaged base, such as a uracil or some other base, the enzymes will then chop out the base by cutting that glycosidic bond. And then you'll come in here to remove that section of the DNA. And then DNA polymerase will come in and start at that nicked site or that broken site start polymerizing, and then DNA ligase, will ligate those last two bases together. And there are even more proteins and enzymes involved in this process that I'll show in a few more minutes. That it's not just a few enzymes and proteins involved. Many enzymes and proteins are involved in this. If any one of those enzymes is also mutated or non-functional, that will decrease the probability of repair for that mutation. And here's the general process of nucleotide excision repair, and in this case having a thymine or thymidine dimer. It distorts the DNA, so there are proteins that run along here and recognize when there's a distortion like that. That whole piece is chopped out, and much more is actually chopped out by an endonuclease. And then DNA polymerase then copies that strand or makes a new strand complementary to the old strand, and then DNA ligase finally puts together the last two nucleotides with a covalent phosphodiester bond. So as I indicated before, there are people where this particular repair pathway has one or more mutated proteins in it, so it doesn't function. And this is a disease called xeroderma pigmentosum. When these people go out into the sun or are watching television or in a fluorescent light or anything that contains even a small amount of UV irradiation, then it generates thymidine dimers. It does the same in you also. But their repair systems don't correct those. So you're lucky enough that you don't have that problem. And even if you do go out into the sun, these enzymes repair those lesions. When they're not repaired, such as in these people, many of those lesions can result in cancers, in skin cancer. So it's a pretty dangerous disease. They have developed ways where these people can actually get out in the sun by having UV protective sunscreens and things like that they can wear so that they can go out into the sun at least for a short time, not for extended periods of time. So this shows some of the molecular mechanisms and proteins involved in this repair system. So as I said, there's a lot more proteins involved in this than I've indicated in those simple diagrams before. So here up at the top, if you have UV damage, then you can go through transcriptional coupled repair of that, essentially or eventually cutting out that whole section with the thymidine dimer, and then going through DNA polymerase to repair that piece. There's also global genomic repair system here that will again recognize and attach to that region with the lesion, cut it apart with endonucleases, and then come back in and repair it by doing DNA replication with DNA polymerase and ligase. If you can see some of the names of these proteins, they say XP, XP, XP. So everything with an XP, those are proteins that have been recognized as being mutated, at least one or more of those proteins mutated in the people with xeroderma pigmentosum, that disease. So you can see there are many potential proteins in there that when mutated cause that disease. So it's not just a single lesion in a single protein that causes a disease. There are many forms then of xeroderma pigmentosum by mutating one or more of those genes that form one or more mutant proteins in that excision repair system. And this is true of all of those repair systems. There are many more than just single proteins involved in those systems, so that if you have one or more of those proteins being mutated or non-functional, then that repair pathway can fail or will fail in most cases. So here again are each one of those that I've talked about, at least in some detail. There's direct reversal, there's the mismatch repair, nucleotide excision repair, base excision repair. And all these excision repairs require endonucleases or glycosylases to cut out the bases or cut out a number of nucleotides. Then there's homologous recombination repair and non-homologous end joining. So there are certain cases where non-homologous regions are joined together in an attempt to correct the mutation. There's also another one over here that occurs in bacteria, archaea, plants, fungi, and some animals. It's not in humans. And this is a photolyase system. And so if you have such as a pyrimidine dimer, this complex recognizes that dimer. It's actually activated by light, so this one's light activated, and then it will repair that region by itself without using DNA polymerase, so it's a much faster system. And you can imagine the organisms that are exposed to a lot of sunlight, as plants are, and some fungi, that this would be a system that would be very efficient at changing, at dealing with pyrimidine dimers. And one more detail about this non-homologous end joining system in bacteria, I think this one is. So if you have all of these proteins, and there are several proteins involved, then you have a double-stranded break, and that can be resealed by this system. If these two proteins are mutated, so those aren't available, then you do get some end joining of those chromosomes, but it's very inefficient and sometimes inaccurate at putting things back together. On the other hand, if you are missing these three proteins over here in green, then it's inefficient, but it's mainly accurate. So things are lined up correctly and you get ligation, but not very often. And then if you are missing that Lig4 protein, then it's inefficient and sometimes accurate, sometimes inaccurate. So you can kind of see that the function of some of these is to recognize the lesion, to bring the two strands together, and then to ligate them together accurately. And so they all have to participate together to do that efficiently and accurately. And again, each one of these repair systems requires more than one protein, and in many cases, several dozen proteins in order to perform that function, and especially the ones that cut out an entire section. They also depend on DNA polymerase and DNA ligase, each of which contain multiple subunits, to come back in there and to build the new strand that has been first mutated and then chopped out by the repair system. So finally, again, to bring this back to you, again, there are 10 trillion cells in your body, each of which are the product of replication cycles. And if there's on average one mutation per genome per replication cycle, then and that 1% of your genome encodes proteins, and therefore there will be approximately 10 to the 13th mutations in genes generated in your cells during your lifetime. Roughly half of those will be non-synonymous mutations in those mRNA encoding genes, and half of those will cause a noticeable change in the proteins. So that means throughout your body, about 6,000 of your 23,000 genes will have at least one mutation in one cell, not in all cells, just in one cell here or there. If the mutation rate is 10 per replication cycle, then there would be 60,000 instead of 6,000. So if you have a problem in your repair enzymes, then you'll generate even more mutations. And if it occurs earlier in your development, then there will be even more mutations in specific genes. And of course, much more of your genome consists of transcribed small RNAs that control expression. So more mutations would be expected in those. So the implication then is also that no two cells in your body have exactly the same genomic sequence. They're different, and so there's sort of evolution of a different kind going on in your body, and hopefully none of that will lead to deleterious mutations in one part of your body or another, but it's very possible. So mutations are a normal part of living organisms, and mutations have been responsible for creating genetic diversity, genetic mutations, and eventually evolution, selection, and evolution. So they're absolutely necessary in any biological system. However, they do cause some problems, and we'll talk about that a little in a subsequent lecture when I talk about cancer. Okay, so that's all I have to say about mutation and repair, except for recombination, which I haven't gone into detail here. But in a subsequent video, I'll talk about the details of recombination, both in repair and in other functions in biological systems. Okay, so I will talk to you later. Thank you.